So today we will talk about the thyroid hormone. As we know that thyroid hormone is synthesized within the thyroid gland. So it is very important that first we discuss about the structure of thyroid gland, then we will move into the synthesis of thyroid hormone. So thyroid gland is located immediately below the larynx on the each side of and anterior to trachea, and it is one of the largest endocrine gland in our body. Now within the thyroid gland, we have a structure which is known as follicle. Now the follicles are filled with follicles. These are the round structures shown in the image. These are lined by cuboidal epithelial cells, and within these enclosed cell we have specialized secretory substance which is known as colloid. And in short, we can say that colloid is a substance present inside the follicle, and that colloid is lined by cuboidal epithelial cells. The major component of colloid is a large glycoprotein which is known as thyroglobulin, and the thyroglobulin contains the thyroid hormones. Now this is a brief overview about the follicle. Now we will talk about the synthesis of thyroid hormone. As we know that thyroid hormone is synthesized within the thyroid gland, and we have follicles inside the thyroid gland as we have discussed previously. So the follicles, they are lined by cuboidal epithelial cells, and the synthesis of thyroid hormone takes place in these cells. So the first step in the synthesis of thyroid hormone is the transport of iodide from the blood into the glandular cells or thyroid glandular cells or follicular epithelial cells, which are cuboidal cells. The energy which is required to move the iodide ion from blood into the thyroid follicular cell is derived from sodium potassium ATPase, because we have to move the iodide from an area of low concentration, that is from the blood, into the thyroid follicular cells which is already loaded with the iodide ions. Now thyroid follicular cells, thyroid follicles cells, once they receive the iodide ion, they start concentrating iodide inside them. So the process of concentration of iodine within the follicular cell is known as iodide trapping. And the follicular cells have the ability to concentrate iodide to about 30 times its concentration in the blood. When the thyroid gland becomes maximally active, the concentration ratio can rise to as high as 250 times. So in short, the iodide ion will move from blood into the follicular cell, and it start concentrating inside the follicular cells. The next step is the transport of iodide ion from the follicular cell into the follicular lumen. Now the iodide will move from the follicular cell into the follicular lumen via a transporter or a protein which is known as pentrin. So pentrin is an important protein which is located on the membrane of the follicular cell, and it not only transport iodide ion outside the cell, but it also transport colloid from thyroid lumen into the cell. This is the second step. The third step is the oxidation of iodine into the iodine. So oxidized iodine. So this will take place with the help of an enzyme which is known as peroxidase enzyme. So peroxidase enzyme will convert iodide ion into oxidized form of iodine. The next step is the secretion of a glycoprotein which is known as thyroglobulin from the follicular cell into the follicular lumen. So thyroglobulin is synthesized within the follicular cell, and it is transported out into the follicular lumen, and here the iodination and coupling of thyroglobulin with the iodide ion or oxidized form of iodine occurs. The next step is the combination or coupling of the iodide ion or oxidized form of iodine with the tyrosine molecules. So on the thyroglobulin molecule we have tyrosine. So we can say the thyroglobulin contains tyrosine, and on that tyrosine the iodine ion will bind. So when one iodine ion binds to a tyrosine, this results in the formation of monoiodotyrosine, and it is denoted by MIT. When two molecules of oxidized form of iodine will bind to thyroglobulin, then this results in the formation of diiodotyrosine. So we have monoiodotyrosine and diiodotyrosine. The next step is the combination of monoiodotyrosine with diiodotyrosine, or combination of monoiodotyrosine with monoiodotyrosine or diiodotyrosine with the diiodotyrosine. So when one monoiodotyrosine combines with one diiodotyrosine, it results in the formation of T3, which is known as triiodothyronine. And when two diiodotyrosine molecule binds with each other, combines with each other, this results in the formation of thyroxine, which is also known as T4. So we have T3 and we have T4. Now once the follicular cell is stimulated by thyroid stimulating hormone, this is a stimulus for the secretion of thyroid hormone into the blood. Okay, so what will happen that the thyroglobulin, which is bound with T3 and T4, is internalized into the follicular cells with the help of a process known as pinocytosis. So it is pinching inward of a protein molecule. Once inside the cell, the colloid droplet, which is actually thyroglobulin containing T3, T4, MIT, and DIT, undergoes cleavage by the proteases. So proteases will cleave the colloid droplet into MIT and DIT and T3, T4. The T3, T4, they are eventually transported into the blood, while the monoiodotyrosine and diiodotyrosine, they undergo further cleavage, or they undergo iodination, and they are converted into iodine and tyrosine. The tyrosine can be reused for the formation of thyroglobulin, and iodine can be reused for the synthesis of next thyroid hormone.